Professor David Haigh is a newly appointed visiting professor within the Faculty of Science and Engineering here at Manchester Met. In 1981, David graduated in Natural Sciences Chemistry from the University of Cambridge and then conducted organic chemistry PhD studies in natural product synthesis at the University of Leeds in collaboration with ICI Pharmaceuticals. Now this was then followed by a postdoctoral fellowship in organic chemistry at the University of Nottingham, again in natural product synthesis. In 1986, David was appointed as a medicinal chemist in the diabetes programme at Beecham Pharmaceuticals in Epsom in Surrey, and there he made an important contribution to the discovery of Avandia, which was a novel drug for the treatment of type 2 diabetes. And in recognition of this, he and several colleagues were joint recipients of the Society for Medicines Research Award for Drug Discovery. David then became Assistant Director of Medicinal Chemistry at SmithKline Beecham's Harlow Research Centre in 1999, and during this period he was engaged in other aspects of diabetes research. David was then appointed as a research manager within infectious diseases medicinal chemistry at GlaxoSmithKline's Medicines Research in Stevenage. Here he led research teams in the identification of novel treatments for hepatitis C virus infections, resulting in 2 medicines which entered preclinical and clinical development for HCV. And in recognition for his contribution to chemical research, Professor Haigh was elected as a Fellow of the Royal Society of Chemistry in 2008. David was subsequently appointed as Professor in Medicinal Chemistry at Queen's University Belfast. In this role, he was Head of Drug Discovery and Enabling Technologies within the Centre for Cancer Research and Cell Biology, and he led teams engaged in cancer research. This included programmes to develop novel drugs for the treatment of lung cancer, which was awarded multi-million-pound funding from the Wellcome Trust in 2012, and which has identified exciting prospects for future treatments. Professor Haigh resigned his chair in Belfast in 2013 and relocated to Cheshire to act as an independent drug discovery and medicinal chemistry specialist advisor. And it's in this context that he is now involved with Manchester Metropolitan University. So I'm sure you'll agree with me that he is an amazing role model for all of us here, and we're delighted to welcome David to our team as a visiting professor. And more specifically, we're delighted to welcome David to present his presentation today, The Language of Chemistry in Drug Discovery. So thank you both so much.
Thank you very much for the introduction, and thank you very much for the invitation to come and lecture at Manchester Metropolitan University today. Before I start, I'd just like to get a sense of the composition of the audience tonight. So I'd like to have a show of hands, please, if I could. How many people in the audience consider themselves to have a chemical sciences or a biological sciences training, or indeed a study in chemical or biological sciences? So it's the majority of the audience, but not all of the audience, okay? Well, those of you that understand chemistry, those of you that are studying chemistry, those of you that are studying biological sciences, please bear with me tonight, because what I've done in the interests of trying to make this lecture accessible to everybody is I've actually taken a few liberties with the science, and I'm going to make the science somewhat simpler than it actually is. So if you feel that I'm not quite telling the whole truth with the science, please accept my apologies for that. It's purely in the interests of trying to make the science accessible to non-scientists. And for those of you that are non-scientists, please bear with it. There is chemistry in the lecture, but I don't expect you to try to understand the chemistry in any detail. What I really want you to understand are the concepts that I'm trying to illustrate. And if you go away from here understanding a few of those concepts, then I think I've achieved the aim that I set out to achieve today in making this lecture an accessible lecture for everybody. Now, I've lectured at a number of venues throughout the UK, throughout Europe, and in America. I've lectured to huge audiences, 2,000 plus people, at American Chemical Society conferences when I've presented on brand new molecules that have entered drug development or even launched in the case of Avandia. I've lectured to very small groups of half a dozen senior company executives. I've lectured to distinguished academics when I was trying to persuade the Wellcome Trust to give me £4 million, as it turned out, to work on a lung cancer project. But today's different. And it's different because for the very first time in all of those dozens, hundreds of lectures that I've given, I'm actually proud to have my parents in the audience tonight listening to this lecture. No pressure then. So this one's different, this one's special. Mum and Dad, this one is for you. So how then do we begin to describe the process of drug discovery? Well, I think we can describe drug discovery in this rather eloquent, if not long-winded manner. As we know, there are known knowns. There are things that we know. We also know there are known unknowns. That's to say, we know there are some things that we do not know. But there are also unknown unknowns. The ones we simply don't know, we don't know. And that, in a nutshell, sums up drug discovery, I think. Now, the path to a new medicine is astonishingly complex. You can't read the text on this slide, it's so small, but every line of text on this slide indicates an activity that is required in order to take a new drug from the concept through to a marketed drug. A huge amount of activity across a wide range of scientific disciplines. It's very complicated, as you might imagine. And so because it's so complicated, what I'm going to do today is to focus on just a few of the concepts in the early stage of drug discovery. And I'm going to present this from a chemistry perspective, but remember that drug discovery really is a truly multidisciplinary science. So I'm going to focus then on a little bit about an outline of the drug discovery process. I'm going to give you a brief introduction to some medicinal chemistry concepts and to some concepts about molecular property space. I'm going to give a brief introduction to proteins and target class biology. And then I'm going to pull all of those concepts together in the second half of the talk with an example of the use of medicinal chemistry in a typical drug discovery scenario. So when we think about drug discovery research, we can really break it down into several themes. So biological scientists will have an idea that treating a particular disease might be accomplished by targeting a specific process, a disease process within the body. If that is thought to be a valid idea, then that will pass on to the hands of research scientists, where the actual process of discovering the potential new medicine takes place. So that's the research phase. Beyond that, then there are a variety of additional components to the process of taking a new drug to market, and that includes the technical phase of manufacturing the medicine commercially, the safety testing, obviously very important, the clinical trials which show that the drug is safe and effective in humans, and then a whole raft of regulatory and other commercial activities to lead to a new drug launch. Now, the cost of new drug discovery is exponentially expensive, getting worse with each step you go along the way. And so there's an old saying in drug discovery that is, if you're going to fail, fail early and fail cheaply. And that's particularly important in the context of the industry as a whole. So I've got some data on the slide here, which happens to be taken from the period 1994 to 2003. But the story is essentially the same, continuing beyond that. And that is that if we look at research and development productivity measured by new medical entities, by new drugs reaching the market, that productivity declined by 40% in the period 1994 to 2003. But at the same time, R&D expenditure increased by 70% in that same period. And in fact, over the last 30, 35 years, R&D expenditure has increased by at least 600%. And in 2003, when this data was put together, the estimated cost per drug entering the market was around about $1.4 to $1.7 billion US. Today, 10 years later, in 2013, that price has risen to approximately $2.7 billion for a new drug to enter the market. And if you take into consideration the fact that every drug that doesn't get there also costs money, and you add those costs in, then the real cost of a company bringing a new drug to market works out at around about $5 billion per new drug that actually makes it onto the market. And the overall probability of success of a drug reaching the market is very low. There's about a 1 in 5 chance that a biological idea will translate into a molecule that enters the development phase of the process. And of those molecules that do enter development, about a 1 in 10 chance that the molecule will actually get launched as a commercial medicine at the end of it. Although if you're looking at compounds that act by a mechanism that we understand, so it's a drug that's similar to something that's already on the market, then from first time in man, FTIH, to market, there's about a 1 in 3 chance for a drug that operates via a mechanism that we already understand. So as you can see, there's a huge attrition rate, and it's incredibly expensive. But the rewards for the companies that are involved are obviously significant. They need to be when they're spending that kind of money. And I've got here some data on the top-selling drugs in the United States in 2016 by sales volume. And the number one best-selling drug in the US in 2016 was a compound called Humira. This is an intravenous injection of a monoclonal antibody, so it's a protein-based drug. And it's used in immunological diseases such as rheumatoid arthritis, psoriasis, and inflammatory bowel disease. The number 2 top-selling drug was a compound called Harvoni. This comes from an area that's close to my heart, hepatitis C infection, and this is actually a small molecule combination of 2 drugs used in the treatment of hepatitis C. The number 3 drug is a compound called Revlimid. This is actually, for those older members of the audience, is actually a very close analogue of a drug called thalidomide that gained notoriety in the 1960s because it caused birth defects in children. And so Revlimid sold $6.9 billion US in 2016. If we look at it from a slightly different perspective, then, and we ask the question, which medicines get prescribed the most well-prescribed medicine of all in the US in 2016 was a compound called lisinopril, which is an antihypertensive, lowers blood pressure. Now, this is a chemistry lecture, so I'm afraid we have to start where all chemistry starts, and that's with the periodic table. And for those of you that are not aware of it, then the chemistry of life is really the chemistry of carbon compounds, carbon combined with other elements, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. So hydrogen, nitrogen, oxygen, phosphorus, and sulfur combined with carbon to give us the molecules of life. Organic chemistry, in contrast, is the wider study of the chemistry of all carbon compounds. And medicinal chemistry is the application of organic chemistry in the process of drug discovery. And as a general approximation, drug molecules are largely restricted to compounds of carbon, hydrogen, nitrogen, oxygen, and sulfur, together with the halogens, fluorine, chlorine, bromine, iodine. You notice that phosphorus tends not to be used in drug molecules, even though phosphorus is an important element in the chemistry of life. And that's because most organophosphorus compounds are actually toxic. Now, in the case of the chemistry of life, then a number of metals are used in the process, as I've indicated in the dashed squares. The most important ones being sodium and potassium, which are involved in conduction of nerve impulses. Iron is another important one, sits at the center of the heme molecule, the molecule that transports oxygen around the body in the blood. But in medicinal chemistry, we tend not to use metals in drug molecules. with the possible exception of things like sodium and potassium that are used as salts in order to improve the physical properties of molecules. Now again, apologies to chemists in the audience if you feel that I'm trying to teach you how to suck eggs here. So the specific orientations of atoms bonded together make unique chemical compounds. So for example, if we look at this structure down in the center of the slide here, here I'm depicting carbon, hydrogen, oxygen, chlorine and nitrogen atoms joined together in a particular orientation. And you can see instantly this structure is rather cumbersome. It almost looks like some sort of a porcupine. And so chemists have devised a shorthand notation for chemical structures to actually make chemical structures much easier to interpret. So this structure here is drawn by organic chemists in this much clearer manner here, where we don't explicitly draw every carbon atom. Instead, we imply that every corner on the molecule here, here, here represents a carbon atom, unless we say something else. We don't draw hydrogen atoms at all, unless we particularly want to highlight them, for example, in this NH2 group here. And a lot of the other atoms that are involved in the molecule we call functional groups, and we have a shorthand notation for those. So for example, this particular orientation of carbon, 2 oxygens and a hydrogen here is referred to as a carboxylic acid, and organic chemists have a shorthand notation, CO2H, which makes it a lot easier to read and understand what the molecule is about. Now, the important thing to appreciate about chemical molecules, if you're not a chemist, is that different chemical structures have different chemical and different physical properties and different shapes. So I've drawn the compound here where I've got the carboxylic acid group at the top of the molecule and the amino group at the bottom of the molecule. If I were to swap those two groups around, I'd still have the same atom count, C10, H12, etc., but I'd have a different compound with different chemical and physical properties. And that's really what we need to keep in mind when we think about some of the chemistry that I'm going to describe later. Now, there are lots of different functional groups that are used in drug discovery. I'm just showing you just a few of them here, just to give you some of the concepts that I want you to keep in mind. So at one end of the spectrum, we have what we call charged or ionic groups. So for example, a carboxylic acid group can lose a hydrogen ion, H+, giving a negatively charged carboxylate ion. So we've got charged molecules like carboxylate. Amines, on the other hand, can pick up a proton to give an ammonium species, again a charged species. At the other end of the scale, we've got lipophilic or greasy groups. So simple carbon chains or carbon rings are quite greasy in their nature and represent the opposite end of the spectrum to the charged groups. And in the middle, we have what we call polarized groups. You can think of these groups as being like sort of little magnets. You've got one end of the magnet will attract the opposite end of another magnet. They're not separate species, they're in the same block of iron. But think of them in kind of magnet-type terms. So here we've got polarized bonds. And just like the opposite poles of a magnet attract, then so polarized atoms will attract each other. Opposites attract. And that's an important concept that we'll come back to later. And along with molecular shape, that's the volume and the three-dimensional shape, lipophilicity and polarity are very important concepts in drug discovery. Now, the chemists in the audience will already be thinking that there's significance in the fact that I've drawn this next slide as a tetrahedron. Those of you that are chemists will immediately realize that by drawing a tetrahedron, I'm inferring that simple saturated carbon compounds have a tetrahedral structure, a three-dimensional shape to them. So molecular shape is an important concept to keep in mind, three-dimensional shape as well as volume. We've talked about lipophilicity, we've talked about polarity, but there are also other concepts that I want you to keep in mind. First one is solubility, the ability of a compound to dissolve in liquid, and in the case of drug discovery, the most important liquid is water, because the body is made up of water. And To go along with that concept of solubility, we also need to think about the concept of permeability. That's the ability of a molecule to pass through a biological membrane, to enter into a cell. So molecular volume, molecular shape, lipophilicity, polarity, permeability, solubility. Those are the concepts that I need you to keep your head on as we go through the lecture, and you'll see the relevance of those at various stages in the discovery process. Now, it's not just those concepts. Molecular property space has lots of parameters that it can be described by. You've got other parameters such as efficacy or potency against your desired biological target, the ability to treat the disease that you're interested in treating. You've got toxicity. That's biological activity against a target that you didn't want to hit. You've got drug metabolism, the body's ability to remove and eliminate drug substances. And you've got bioavailability, whether you can actually get enough of your compound into the body to have the desired biological effect. And the bottom line is that understanding these properties is what's going to help you to design a new drug. So there are lots and lots of potential biological targets. Think of them as haystacks. This field is full of haystacks, just as the body's full of biological targets. The trick in this game is to decide which haystack is the one you're going to look in for that drug substance, that needle, and then come up with a very good strategy for searching through the haystack to find the needle once you've decided which haystack you're going to look in. And that really takes us back to this slide that I started with. And really what I want to do for the rest of the talk is to focus on a very small area of the slide. And that's this bit up here, still too small to read. So we look at that in more detail. There's still far too much information on that. And so I'm going to simplify that even further down to this simple schematic cartoon. So here we go then, the drug discovery process. Our DNA contains lots of genes. Each gene codes for a unique protein within the body. So genes codes for proteins. Any one of those proteins could be a potential target for drug discovery, a biological target, a molecule in the body that we want to try to manipulate with a drug substance. So imagine then that we can take our biological target and we can then find a method by which we can test chemicals for their ability to influence the properties of that biological target, some sort of assay system that allows us to measure whether compounds influence that target or not. We call that process screening. If we then take a huge collection of chemicals, drug molecules, and we test that collection of molecules against our biological target, hopefully we'll find a small handful that work against the target that give us the activity that we're interested in. So for example, here I've got this triangular molecule which has got the right shape to fit into the biological target. It's active against the target, but all the other molecules don't work. So that then gives us a small number of compounds which are active against that target. And if we've done that process using tens of thousands, hundreds of thousands, in the case of the GSK compound collection, millions of compounds, we call that process high throughput screening. So we've got a hit, this triangular compound, and the process that we go through then is called the hit to lead and the lead optimization part of the drug discovery process. So we want to be sure that this compound is a genuine compound and that it's a compound that we can actually work with. That lets us go through that sort of process iteratively. We stop calling it a hit, we start calling it a lead molecule once we're happy that it really does what we want it to do. And then the idea is by changing the size and the shape of the molecule, as I've depicted here with these different blobs, to try to find a molecule that has got all of the properties that we want and none of the properties that we don't want. And so we go through an iterative process of making compounds and testing compounds, looking at the data we get back and using that data to make the next generation of compounds and the next and the next and the next, until eventually we get a compound over here that has got the biological properties that we think are important to become a drug substance. So all through that process, we're thinking in terms of biological targets, hits, leads, and when we get one that we think is going to be a good one, in GSK we used to call that a candidate molecule. Other companies have different names, but it's the same stage in the process. Beyond that stage, as you start to move into longer-term animal studies, safety testing, clinical trials, you stop calling them compounds, you start calling them drugs. So that's the point at which we start referring to them as drugs. Prior to that, we think in terms of hits, leads, candidates. Now, there are lots of different types of biological targets that we could think of. Remember, genes code for proteins, which are biological targets. And those biological targets can be grouped together into a number of families based on their similarity to each other. And we call those target classes. So, for example, if we think of steroid drugs act via steroid receptors, and steroid receptors are members of the nuclear receptor family. So these drugs act by influencing the ability of genes to produce proteins. They switch genes on, they switch genes off, they produce proteins or stop the production of proteins within the cell nucleus. There's a group called the 7-transmembrane receptors. These proteins sit on the surface of cells, and when a drug molecule interacts with the receptor sitting on the surface of the cell, it triggers a signaling cascade within the cell. And an example of that would be drugs like Zantac, the anti-ulcer drug, which interacts with the histamine H2 receptor target. Salbutamol, the anti-asthma drug, acts at the beta-2 adrenoceptor. There's a group of targets called proteases. These are proteins whose job is to cleave and destroy other proteins. And an example of a drug that works against a protease would be Amprenavir, which is a drug which is used to treat HIV infection. It inhibits the HIV protease. There are ion channels. For example, lidocaine, the local anesthetic that is used in dentistry, for example, blocks a sodium channel. And sodium is the ion that is responsible for triggering a nerve impulse. So by blocking the sodium channel, you stop the inflow of sodium into the nerve, and you stop the pain, hopefully, although in dentistry I'm not quite sure. Diazepam is an anxiolytic, reduces anxiety. That operates via an ion channel called the GABA channel. And then the other big group is a group called the kinases, and an example of that would be Tykerb, which is an anti-cancer drug. Now again, apologies to the biologists and the biochemists in the audience if you feel that I'm teaching you to suck eggs here. Genes contain the code to make proteins, and proteins are made up from 20 different naturally occurring amino acids, the building blocks of protein. And what I'm showing in this cartoon here is 5 amino acids: leucine, aspartic acid, tyrosine, valine, and proline, bonded together to give this 5 amino acid peptide, this pentapeptide, where each amino acid is joined to the next by this C double bond O NH group. C double bond O NH, C double bond O NH, the peptide bond. Now, proteins are made up of chains of tens, hundreds, thousands of amino acids linked together in this way. And you can think of those as a huge, great long length of string. But then certain types of amino acids in a particular sequence can coil together to give helical structures, like the one I'm showing here, called an alpha helix. Other groups of amino acids linked together can give a sort of a flatter ribbon type structure, which we call beta sheets. And then these secondary structures, these alpha helices and these beta sheets, can themselves conglomerate together to give us the three-dimensional globular structure of the final protein. Now, the picture that I'm showing you here is an X-ray crystal structure of a crystal of a particular kinase. This one happens to be glycogen synthase kinase, and we'll come back to that in a moment. But it illustrates the idea of a long chain of amino acids linked together. Some bits are alpha helices, some bits are beta sheets. And in here, this region, we've got these 5 amino acids linked together here. So how do drug molecules interact with their target proteins then? Well, there are several different types of interaction that we need to think about. The first one is the ionic or the salt interaction. So a charged carboxylate anion of the type that I mentioned earlier can interact with a positively charged amine group on the protein, give a salt bridge, an ionic interaction between positive and negatively charged functional groups. At the other end of the scale, we've got the hydrophobic interactions, the greasy interactions. Everyone's familiar with the idea that if you sprinkle oil on the surface of water, the drops of oil tend to congregate together into a bigger lump of oil. That's a hydrophobic process in action. And so hydrophobic interactions are important in drug discovery. The interaction between the hydrophobic parts of the protein and the hydrophobic parts of the drug molecule give us an interaction that can be important for a drug binding to its target. And then in between, we've got the so-called hydrogen bond interactions between these polarized groups, these little magnets, where the charge is not completely dissociated, as in the case of the ionized groups. You've got these partial charges, think of them like little magnets, and the opposite ends of the magnets attract each other. We call those hydrogen bonds, and that's a very important group of interactions that take place between drugs and their target molecules. There's actually a fourth type of interaction that's important in drug discovery, and that's a covalent interaction, where the drug molecule becomes directly attached to the protein. And that's useful for some drugs, like protease inhibitors, but generally it's not an interaction that you particularly want because it's often associated with toxicity. So we tend not to use covalent interactions unless we're thinking in terms of particular types of targets like protease inhibitors. Okay, so that's all the background science that I really want to bring to the table today. What I would like to do in the second half of the talk now then is to give you an illustration of how we use those concepts to operate a drug discovery program and to design molecules that will interact with a specific biological target. So I'm going to talk about an anti-diabetic research project that I was involved in a few years ago now. And we'll start by telling you a little bit about diabetes so that you understand what we were trying to achieve. So type 2 diabetes is a disease that's caused by loss of control of glucose use and glucose storage. And so type 2 diabetics have elevated blood glucose. And that sort of elevated blood glucose is underpinned by a body having a reduced response to insulin. And insulin is the hormone that controls blood glucose. So in a normal, healthy individual, a lean individual, in response to a meal, glucose rises in your body. The body then secretes insulin. Insulin acts as its target tissues, reduces blood glucose back to normal. Everything's okay. As people become obese, then the body is starting to struggle a little bit to maintain that control, but the control is still there. But as the disease worsens, you reach a state called impaired glucose tolerance, where now the body is really struggling. It's only just managing to keep control of blood glucose, and it's doing that by producing massive quantities of insulin in order to try and keep control of glucose. And eventually you reach the stage where the body simply can't produce enough insulin and where the insulin just isn't able to give you the desired effect anymore. That then leads to loss of control of glucose and you become a type 2 diabetic. And type 2 diabetes is an important disease in its own right, but it's also fundamental to the long-term development of a whole variety of other diseases: heart disease, stroke, kidney disease, nerve disease, diseases of the eye, etc. So diabetes is a really big problem, but fundamentally it's a disease of loss of control of blood glucose. Now, when I started in diabetes research back in the early 80s, we knew a tremendous amount about how insulin worked, and it's indicated on the slide here. We knew absolutely nothing other than that insulin was the hormone that controlled blood glucose. And so in order to test compounds in those days, we used an animal model. The animal model was an obese mouse. So here is a lean mouse. Here is its obese offspring. And these obese mice, these were genetically obese mice, and they represented a good model for testing compounds for the treatment of diabetes. We could give anti-diabetic compounds to an obese mouse and look at how the compound affected the blood glucose levels within the mouse. So we tested compounds in an obese mouse. We didn't know what the target was. We had no idea what the mechanism of action was. What we knew was that compounds worked in obese mice. And from that, we were able to come up, in the case of SmithKline-Beecham at the time, with the drug Avandia that Liz mentioned in the introduction. Now, things have moved on, of course, since then, and this slide was prepared a few years ago, and even this slide is out of date now. We now know a tremendous amount about what happens when insulin acts at an insulin-responsive cell, such as a liver cell, a muscle cell, a fat cell. There's a huge number of biochemical processes that are going on inside the cell that act to maintain the control of blood glucose within that cell. And I just want to focus on one particular process, and that's the process of glycogen synthesis. And glycogen synthesis is controlled by an enzyme called glycogen synthase, which in turn is controlled by a kinase, glycogen synthase kinase. So we'll come back to that in a moment. But first of all, we perhaps ought to say a little bit more about what kinases are. So kinases are phosphate transfer enzymes. These proteins, these enzymes, transfer a phosphate group from adenine triphosphate, ATP. They put a phosphate group on a protein. So what? Well, by transferring a phosphate group onto a protein, you change the shape of the protein, you change the activity of the protein. And so that's the way in which the body regulates some of these signaling cascades, by putting phosphate groups onto proteins. And they do that by phosphorylating, adding a phosphate group, to particular amino acids within the protein, amino acids that contain an OH group. Three amino acids contain an OH group: serine, which I'm showing in the slide here. There are two others, threonine and tyrosine. All of those contain an OH group. All of those can be phosphorylated. All of those are phosphorylated by certain kinases. So kinases transfer a phosphate onto a protein. So here then is glycogen synthase kinase. Here's the X-ray crystal structure that I showed you previously. And here on the left-hand side of the slide, imagine the sort of a computer-modeled protein surface. So this is, if you like, what the surface of the protein might look like if we could get a really powerful microscope and look at the actual surface of the protein. And the important thing to take from this is that the molecule ATP binds into a binding site, into a groove in the surface of the kinase enzyme. And then the terminal phosphate group is this one here. That's the one that is going to get transferred onto the protein that is going to get phosphorylated in this process. And glycogen synthase kinase is important not only in diabetes, but also in neurological disorders, and increasingly GSK3 is becoming quite relevant in the treatment of cancer. So what does glycogen synthase kinase do? Well, let's just recap on that. So the important thing to remember is that we want to reduce blood glucose. And one mechanism by which we can reduce blood glucose is to convert glucose into glycogen within the cell. That process, as I've said, is controlled by glycogen synthase. But GSK3 puts a phosphate on glycogen synthase and switches it off. So glycogen synthase kinase is actually a bad enzyme in the context of controlling blood glucose. And so the concept then was that because glycogen synthase kinase levels are elevated in diabetes, with a corresponding reduction in the body's ability to synthesize glycogen, if we could block glycogen synthase kinase, what we would be able to do then would be to keep the activity of glycogen synthase, and by keeping the activity of glycogen synthase, we'd convert more glucose into glycogen, and by doing that, we'd be able to lower blood glucose and have a potential anti-diabetic drug. So that was the concept of why we were interested in studying glycogen synthase kinase as an anti-diabetic approach. Now, we had a couple of methods by which we could measure how active compounds were. We had a simple enzyme assay measuring the activity of the kinase enzyme, And we did that using a small fragment of the target protein, glycogen synthase, 27 amino acid fragment, and we could measure the incorporation of phosphates into that using radioactive phosphorus. We also had a cellular assay, which was giving us an estimation of how active the compounds were in a cellular context. Remember, all these processes are taking place within cells within the body, so we want to have a cellular assay. In this case, we were looking at the incorporation of radioactive glucose into glycogen in a cellular context. And for those of you that are not familiar with units, we'll talk about IC50, the inhibitory concentration required to inhibit 50% of the enzyme activity. We'll also talk about EC50, the concentration of a compound needed to give 50% of the maximum effect in a cell. There are also the units that are involved here, micromoles and nanomoles. 1 micromole is 1,000 nanomoles. So we carried out a high throughput screen of the SmithKline-Beecham, as it was then, compound collection. And we got a handful of hits came out of that process, one of which I'm showing you here. And when I was looking at the screening output, this particular molecule struck me as being quite an interesting proposition. Think back to the different shapes that I showed you interacting with the target. Think of this as being the triangle, the one that looked quite interesting. Why was it interesting to us? Well, it's cell permeable, it gets into cells, and that's going to be important. But it also had reasonable potency and selectivity for the inhibition of GSK3. It was ATP competitive, and I'll come back to what that means in a minute. But more importantly, from a structural point of view, this was the only compound we had in the SmithKline-Beecham compound collection. Hundreds of thousands of compounds in the SmithKline-Beecham compound collection, but we only had one of these, this one. So that was interesting. And it was even more interesting when I looked in the chemical literature and discovered that it was a very, very poorly exemplified molecule in the chemical literature. Okay, so we don't know anything about these kind of molecules because there aren't many of them reported in the literature. But the important thing for drug discovery is that means that we can make novel compounds that nobody else has made, nobody else has thought about, and therefore we can get intellectual property on these compounds. We can take out a patent to protect these compounds, and that's going to be important for giving us commercial exclusivity to sell the drug at the end of the day. So this looked like quite an interesting chemical scaffold, and you can imagine that if we put different substituents into this ring and into this ring, we can make a whole range of molecules that might have different shape and different properties. Now, I said that the compound was competitive with ATP. What does that mean? Well, here I'm showing you in this cartoon ATP in blue in a sort of a schematic of the kinase binding pocket, this groove in the GSK3 protein that I mentioned earlier. And this is the pentapeptide that I showed you a few slides ago, Lu, Asp, Tire, Val, Pro, this pentapeptide that I used as the illustration of how amino acids link together. And the important thing to notice from this slide is that ATP, via its amino group and via this nitrogen in this six-membered ring, makes 2 hydrogen bonds, these magnets attracting each other, makes 2 hydrogen bonds to this particular region of the kinase enzyme. So that represents the ATP binding pocket, and now how ATP binds into that binding pocket. Now, if you watch this region of the slide for the next few seconds, what you'll see then is that our computer models suggested that this molecule that I showed you a couple of slides ago was capable of mimicking ATP in the binding site. And by mimicking ATP, it's competing with ATP for that binding site. So that's what I mean when I say that the compound is competitive with ATP. But it's more important than that, because if you look at this binding site, there's a lot of white space here. Well, it's not just simply white space. There's a region here that accommodates these phosphate groups, likes to have polar groups sitting there. There's a region here that will accommodate greasy residues, and then there's the core of the molecule. And you can see that with the high throughput screening hit, we're only really occupying the core region of the binding site. So there's the potential to add other groups onto the rings that might give us access to these polar regions and these lipophilic regions. So we're going to use polarity and lipophilicity to try to make more active compounds. Now, I don't need to go into the synthesis of these compounds in any detail, and in fact, the chemists in the audience will have already assimilated the information that's on this slide. Suffice it to say that it's quite easy to make molecules in the blue box here, in which we've got this 6-membered ring connected to our 5-membered ring, and we've got a chlorine atom at this position. And this particular chlorine atom is quite a reactive chlorine atom. And we can react that with a group of amines connected to another 6-membered ring, so-called anilines, and the aniline nitrogen will displace the chlorine to give us the final compound. Very easy chemistry to do. And so you can imagine that if we put different substituents into the left-hand 6-membered ring and into the right-hand 6-membered ring, we can very quickly make a whole range of compounds with different molecular properties, different shape, different polarity, different lipophilicity. And we could do that robotically. And this is the robot that we used. And for the chemists in the audience, this robot conducts 48 chemical reactions simultaneously. It can do liquid dispensing, it can do mixing, it can do heating, it can do cooling, it can even do liquid-liquid extractions, aqueous workups. And we can combine that then with automated purification by a technique known as chromatography, which we could do 10 reactions, or 10 purifications, sorry, at a time. So we could very quickly make large numbers of compounds. This is high-throughput combinatorial chemistry, an expression you might have heard. So imagine then, if you will, that here's our active chloro compound, where the chlorine is represented by this green blob. We can have lots of different substituents in this ring, so lots of different molecules, different pinks. We can react those with different molecules with substituents in this ring, lots of different yellows, and we get the target compound out. And you can imagine that we could take, in our 48-membered reactor, 3 pinks and 16 yellows, 6 pinks and 8 yellows, 4 and 12, etc., etc. We could do lots of different combinations as we explored which of the substituents were the best substituents in the left-hand ring, which of the substituents were the best substituents in the right-hand ring. And we use that information iteratively to design the next set of compounds. And by doing that, we were very quickly able to make well over 1,600 compounds in the space of probably 3 or 4 months, I think, if I remember correctly. Now, I'm not going to show you all 1,600 of those compounds, although I'm sure chemists in the audience would like to see all of those. I just want to illustrate the principle. And so here's the starting point. This compound had a potency, an IC50 of 800 nanomolar. By adding a chlorine substituent into this ring and a carboxylic acid substituent into this ring, we can get about a 5-fold improvement in potency. The number's getting smaller, that means it's more active. We can add an extra chlorine into that ring there, gives us another increase in potency. We can swap this chlorine for a fluorine atom, gives us another increase in potency. So you see that very quickly we've gone from 800 nanomolar to 20 nanomolar, a compound that's 40 times more potent, 40 times more active. And you need to be careful with this kind of biological data. Don't assume that because that says 35 and this says 20, that there's a real difference. If you test those compounds enough times head-to-head against each other, they might turn out to be the same, or they might turn out to be different. So we tend not to think in terms of absolute numbers in this part of the process. What we're thinking of is the trends. So it's safe to say that the one in the bottom right is 40 times more potent than the one top left, but I wouldn't want to draw any inference between bottom left and bottom right. They're of the same order of magnitude, is the expression that you'll hear chemists using. And this is structure-activity relationships. This is looking at how changes in structure influence the biological properties of the molecule. Now, I'm just going to illustrate 3 compounds, 3 different compounds, not the ones that were on the previous slide, but 3 other compounds from this series. And I'm trying to show pictures here of those compounds bound into the ATP binding site on the kinase. So in the first compound, we've got a pair of fluorines in the left-hand ring. We've got another pair of fluorines in the right-hand ring. That molecule binds into the ATP region as shown there. And in fact, The orientation here is flipped relative to what I've drawn it on the slide. So this ring here is that ring, and this ring here is that ring. These white blobs represent the fluorines, so they're adjacent to each other there, that ring, and they're one atom apart here, that ring. Okay? Here's another molecule in which we've added a second ring. We've fused a second ring on. Gives us a molecule like that. We've gone from 80 nanomolar to 19 nanomolar, fourfold increase in activity. And that's achieved because this molecule is more lipophilic. This ring is more lipophilic, so it makes better lipophilic contacts with the protein behind the molecule. It's filling that lipophilic pocket that I told you about. So it's become more potent because we're getting a better interaction into the lipophilic pocket. Here's another one. This one's got a carboxylic acid at the end of it, so it's making a charged interaction, one of these salt interactions with a residue in the front of the molecule. For the chemists, it's an arginine residue. It's an amino-containing group. And so that, again, has given us another increase in potency, 11 nanomolar. So we've got a compound that's making hydrophobic interactions on the left. We've got a compound that's got improved hydrophobic, improved lipophilic contact in the middle. And we've got another compound on the right-hand side of the slide that is using this polar pocket and making charged interactions. And so lipophilicity and polarity are the important concepts that we're using in order to improve the activity of the compounds. Now, those data that I showed on the slide a moment ago were data against the enzyme itself in an isolated enzyme assay. On top of that, we've also looked at these compounds in a cellular assay. And you can see that the first two compounds have comparable cellular activity, 0.47 micromolar, 0.35 micromolar. But this compound on the right-hand side, despite having the best enzyme activity, is actually inactive in our cell assay. What's going on? Well, this carboxylic acid, you remember, is polar, it's charged, and charged molecules like this don't pass easily through biological membranes. So because it can't get through the cell membrane, it can't get into the cell, and therefore it doesn't work in the cell. So polarity is good for making the molecule more potent, but it's a bad thing because the molecule can't get into cells, and we need it to get into cells. So how are we going to address that problem? Well, you remember this tetrahedron that I showed you earlier with lipophilicity and polarity on it? Now we need to think about solubility and permeability. If we want to get a compound into cells, we want to improve cellular permeability. How do we do that? We remove ionizable groups. Well, that's not going to do the potency any good because we need that ionizable group for potency. We reduce hydrogen bonding. We reduce the polarity. We increase the lipophilicity, the greasiness of the molecule. So that's what we need to do to get cell permeability. But on the other side of the equation is solubility. If we want to make compounds more soluble, more water soluble in particular, we need to add ionizable groups. We need to add hydrogen bonding. We need to reduce the lipophilicity. So what we've got to do as medicinal chemists is balance those properties in order to get a compound that's potent against the enzyme, potent in cells, soluble. It can be a tough challenge. Sometimes all these properties work in different directions to each other. But if we're lucky, we can find ways of doing that, and we can get compounds that are active in cells. They've got the right solubility, the right permeability to get into cells and to work into cells. So we've got compounds that work against the enzyme. We've got compounds that work in cells. Now what? Well, this is where drug discovery enters a whole new form of black magic, and that's drug metabolism and pharmacokinetics. Drug metabolism and pharmacokinetics, DMPK, can be thought of as what the body does to the drug. So here's my healthy human volunteer going to take part in a clinical trial. The industry is very good at making compounds that work in clinical trials against healthy volunteers, but I've just realized why they're not so good at treating patients, because our clinical trial volunteers get less and less patient-looking with each passing day. As you can see here, this guy looks nothing like a real patient anymore. So what's going on during the process of getting a drug substance into the body? Well, we give a drug orally by mouth, that drug will pass into the stomach. And when it gets into the stomach's quite acidic, pH 1, hydrochloric acid. And so the drug, first of all, has to be stable to the acidity of the stomach. Some drugs are, some drugs aren't. If the drug's not stable to the acid, it'll get destroyed in the acid in the stomach, and that's that. End of drug. Drugs that are stable to stomach acid then have to be absorbed. And that absorption takes place from the small intestine. Drugs are absorbed through the intestinal wall. They need to be capable of passing through the intestinal wall. And if they succeed in doing that, then they enter the portal vein, and the portal vein takes compounds directly to the liver. It's like playing Monopoly. Do not pass go, do not collect £200, go straight to the liver. That's what happens when compounds get absorbed from the intestine. Everything goes to the liver. So you've got a process here called absorption, which is dependent upon the compounds being soluble and dependent upon them having the permeability to be able to pass through the membranes of the intestine. There are certain drugs that can make use of another feature of the intestinal wall, which is called active transport, but I'm not going to talk about that today. Once compounds get into the liver, then a whole new set of factors come into play, and that's metabolism. The liver is absolutely chock full of enzymes whose sole purpose is to destroy drug molecules. Metabolize those drug molecules, get rid of those drug molecules from the body. That's what the liver's there for. That's what it does all day, every day. And so any compound that you give orally, before it can get into the bloodstream, has to go through the liver. Some molecules will get through the liver, some molecules won't. Some molecules will be metabolized in the liver and then thrown out through the bile, into the bile duct, back into the intestine and excreted with the feces. Sometimes the unmetabolized drug gets excreted that way. But usually it's metabolites that get put back into the intestine and out through the feces. Other drugs will survive metabolism or will get metabolized and will get into the circulation, and they'll get excreted through the kidneys in the urine. So your drug is either going to come out in the feces or the urine untouched, or it's going to get metabolized and come out through the feces or the urine. So you can see that this process of getting a drug into the bloodstream is quite difficult because you've got to get past the liver. But if you can get into the bloodstream, then the bloodstream will distribute the drug throughout the body, and hopefully you'll then get the drug to your target tissue. In the case of diabetes, you need to get it to a liver cell or a muscle cell within the body. And once it gets to the target tissue, then hopefully you'll get the desired biological effect, the pharmacodynamic effect that you're looking for. But of course, it's not quite as simple as that. Nothing ever is, because your drug might also get distributed to other tissues. You might have what's called off-target effects. That's biological activity, but against a different target within the body, a different organ within the body, something that you don't want. So you've got off-target toxicity to think about. You've also got protein binding. Drugs get absorbed onto other proteins. That can be a good thing because it means that the drug is available for a long time in the body, but it can also be a bad thing because the drug just gets stuck on the protein and isn't available in the circulation to give you the desired effect. So you've got a lot of things going on here: absorption, distribution, metabolism, elimination. All of these factors are operating against you in the context of drug discovery. And there are lots of parameters that we can talk about in drug metabolism terms. Some of the ones you'll hear more commonly are bioavailability, how much of an oral dose actually reaches the circulation, half-life, the rate of decay of the compound in the blood, clearance, the ability of the body to metabolize compounds, and exposure or area under the curve, kind of a measure of the quantity of compound that's in the bloodstream over time. And you might ask the question, well, why are some drugs given orally but other drugs given intravenously? The answer is actually quite simple. If you want to avoid gut stability, you want to avoid problems with absorption, you want to avoid first-pass metabolism, you give the compound intravenously. So if you've got a compound that's not going to be stable to stomach acid, you can't dose it through the stomach unless you take other precautions, which I'm not going to go into today. The problem with intravenous dosing, of course, is that it's much more difficult to do, other than type 1 insulin-dependent diabetics who are very used to injecting themselves and drug addicts who inject themselves for fun. Most patients are not very good at self-administering injections, so intravenous drug administration tends to be reserved for hospital use rather than in the general population. So you can see we've got a lot of factors that we need to think about and that we need to bring together in order to have a successful drug discovery program. And that's just to get to the point where we think we've got a compound that might be useful as a medicine. So I just wanted to round that off by giving you some illustrations of how we can manipulate molecular properties to get those factors built in. So here are the compounds that I've shown you previously. That was the screening hit that we started with. You can see 800 nanomolar enzyme potency, not very active in the cell, poor solubility. This was this 80 nanomolar compound with 2 fluorines in each ring, 80 nanomolar against the enzyme, half micromolar in the cells. Okay solubility, but it gave a good oral exposure and had low clearance. Number of 1 is a good number. Low clearance in a rat model of DMPK. Here's our more lipophilic compound. You can see it was more potent. C was more potent than B, but the cellular activity was pretty much the same. This compound had poor solubility and higher clearance, and so you get poor oral exposure. It's not going to make it as a drug because you're just not getting enough of the drug into the body. Despite the fact that it's more lipophilic, more permeable, the downside of this compound is it's not got the solubility. And then here's another compound that I've added to the list. This one now does have a carboxylic acid on it, and I told you earlier that carboxylic acids were no good because they don't pass through biological membranes. But if we balance polarity and lipophilicity, we can get carboxylic acids to pass through biological membranes. And here, by adding another benzene ring, we've made the molecule more lipophilic, and so this molecule can now pass through biological membranes. And you see we've got good potency in the enzyme, good potency in the cell. We've got good solubility because this polar group gives us the solubility that we need. And we've also got quite a reasonable exposure of the drug substance in the circulation. So by manipulating polarity and lipophilicity, we can find molecules that are permeable and soluble. Okay, this is the last of the data slides, almost at the end of the presentation now. So from all of that work, this is the compound that emerged as the best overall compound for future work. So you can see this compound has got good acid stability. Half-life in the stomach acid is over 200 hours. Well, transit time through the stomach is a matter of minutes, so that's okay. It's reasonable solubility. It's got high permeability, so it's going to get into the portal vein. It's going to get distributed to the cells. It's going to get into the cells. It's got good activity profile, as we talked about earlier. And then in the rat DMPK studies, we have oral bioavailability of 65%. So this has everything that we need to potentially go forward as a new medicine, in this case for the treatment of diabetes. Now, unfortunately, it happened that at this time, GlaxoSmithKline reprioritized its research programs, and this particular chemical series was discontinued, so I can't tell you any more about that story. That was factors that were beyond our control. But this work was quite important because it's influenced other groups that have followed on behind us. So we published 3 papers in this series of compounds, and you can see the number of times those papers have been referenced by other authors working in the field of GSK3 research. So we've had almost 1,000 citations of our work by other authors working on compounds for the inhibition of glycogen synthase kinase. So clearly an important contribution to the knowledge in this research area. And as I mentioned right at the start of this section of the talk, GSK3 is not just important for diabetes, it's also important for CNS disorders, and also more recently it's gaining interest as a potential target for the treatment of cancer. Here is a compound from AstraZeneca, AZD1080, and this is a GSK3 inhibitor that's believed to have been in development for the treatment of Alzheimer's disease, although I wasn't able to find any information to tell me whether it's still in development or not. If you look at the picture of this molecule bound into the ATP binding site, the molecule is depicted in a different way, but you can, with a bit of imagination, realize that it's occupying the same space in the binding site that our compound, SB410111, occupies. So it's working in a similar manner. It's using the hydrogen bonds between this NH and this OH to bind to the backbone of the protein, making those interactions, and then it's filling the space in the molecule, and we've got a solubilizing, a polar group on the end of the molecule to make the molecule water soluble and to give it increased potency. So hopefully that's an example of how you can bring all of these things together. And what I really want to leave you with as a final thought then is the language of chemistry in drug discovery, which is the title I gave to the slide. The true language of chemistry in drug discovery is that of understanding how molecular structure influences molecular shape and also influences the physical-chemical properties that are the essential drivers of medicinal chemistry strategy. And with that, I'd like to acknowledge the huge teams of scientists from all disciplines that I've had the privilege of working with and leading over the years during my time in the industry and also when I was working at Queen's University, and to thank you for your interest. Thank you.
Beautifully elegant presentation. Very much indeed. Have you got any questions? Yes. Just to introduce yourself while you give me
My name's Mark Leach. I'm teaching medicinal chemistry to the third and fourth year chemists, along with my Lewis. I showed my students the 200 top pharmaceuticals of those posters. It was in 2009. The top 6 were small molecules. Yes. It was one large molecule, and the rest were now. To the 2016, I think 5 of the 6 were large molecules.
Yeah, the monoclonals.
In 10 years, 20 years' time, are there going to be any small molecules, or is it going to be large molecules?
I honestly don't know the answer to that question. I think as we understand more and more about how to target monoclonals in an effective way and how to turn them into drugs, then we might well see more and more of them coming to the fore. I think there'll always be a place for small molecules.
So as a chemist, I'm referring to small molecules. What about the cost of development of the two? Is there? I know that it's often the price. Normally when we see the NICE recommend things to the NHS, it seems to be it's the large molecules which just have these astonishingly expensive 100,000.
Yeah, I don't know how much the development costs are particularly different. I mean, the regulatory side of the equation is going to be pretty much the same. The clinical trials side of the equation, assuming a comparable patient pool, is going to be pretty much the same. Whether there's any fundamental difference in the research side of developing a monoclonal versus developing a small molecule, or discovering it.
You obviously will be in small molecules.
Yeah, I mean, I can't imagine that it's actually going to make a huge impact because, you know, as I said earlier on, if you're going to fail early. The research phase is cheap. It's the development costs that carry the expense. Right. So I don't know that it necessarily makes a huge difference to the overall cost of bringing a molecule to market.
My name is John. I'm a master's student in organic chemistry. My question is generally, when you're talking about drug patenting, when you're patenting these molecules, in a way, is that not inhibiting the scientific process? These, like, other people cannot use these compounds; they have to come up with analogues and other things like that. Isn't that kind of like contradictory to what we try and do as scientists in progressing forward?
I think the answer to that question is twofold. It is and it isn't. It is in the sense of you're trying to gain exclusivity for your particular company to be the only company that can use that particular molecule. But the very nature of being granted a patent is that you're actually telling the world about your invention. So you're making that knowledge available to everybody, but they just can't use the particular molecules that you've invented. But there's nothing to stop them coming up with a molecule that is very similar, but which lies outside of your patent, based on the knowledge that is contained within your patent. And in fact, if you take that as a concept, the work that I was involved in that led to the discovery of Avandia, there were other compounds that were similar in structure that we used as a starting point for our work.
Well, then isn't it ultimately kind of pointless? You go through all this rigmarole of getting patents and stuff like that when there's compounds that are exactly the same by just the only difference is one bond. So.
Well, I think the answer to that question lies in the cost of developing a drug. Nobody's going to put 2.75 billion whatever dollars into drug discovery if they can't recover that cost. And so what the patent does is it gives you that exclusivity for a limited period of time to sell your compound. It's not forever. It's a limited period of time. It's 21 years from the date of filing, but it can take you 12 years to actually get to market. So the actual period of exclusivity is actually quite small. Might only be 5 years, 10 years, depending upon how long it takes you to get to market. And at the end of that period, then anybody can make that drug and can sell it. And that's what's known as drugs going off patent. So when drugs are first marketed, they have a brand name, Avandia, Humina, I can't remember what the others were on that slide earlier. That's the branded name of the drug. When the drug goes off patent, then anybody can make and sell that drug. All they have to do is to satisfy the regulatory authorities that their version of the drug is biologically equivalent to your version of the drug, and then they can sell it. The drug is then said to be off patent, and then you'll see it being referred to by a different name. So if I just... okay, so you see each of these compounds has got two names. So the first one, Humira brackets Adlamubab, can't pronounce it. Second one, Harvoni, Ledipasvir and Sofosbuvir. Yeah? So why have we got two names? Well, all drugs have two names. The first one, the Harvoni, is the branded name, but it also has a second name, what's known as the generic name. And when the compound goes off patent, anybody else that makes and sells the drug, in the case of Harvoni, because I can pronounce that, they can't call it Harvoni, they have to call it by its generic name. Only the original manufacturer can call it Harvoni. So as drugs go off patent, anybody can make them, the cost comes down, they become a lot cheaper, but you then get these generic copies. Yeah.
So, very quick follow-up. When you say that a drug becomes known as a candidate, is that the moment when it's patented, after it's patented?
It can be patented at any stage in the process. Some companies have a strategy to file early. So the minute you think you've got something, file a big broad claim that protects as much of the area as possible. You may get that patent granted in its broadest sense. You may find that the patent examiners say, we're not going to give you that because you haven't exemplified that, but we will give you this bit because you've exemplified this bit. And so what you get granted might be a lot smaller than what you claim in the initial patent. So some companies will have a file early strategy. Other companies will have a file, what they call a file late strategy. So they'll wait until they've got the molecule and they just file a patent to that one molecule. So that means that you've got a risk that other companies can come along and put a methyl group on and get a new compound, a new patent. So that's a risk of filing late, but by filing late, you extend your patent life.
Which one do you prefer?
In the work that I was involved in at GSK, we had a file early strategy. Whether that's still the case, I don't know.
We've got time for one more question, but there's also opportunity to have a talk after with the refreshments. One more question?
Yeah. Well, David knows me, but I'm Liz. I teach pharmaceutical chemistry here. We have some of our students in the audience. Yeah. And let's say you come straight off your MChem as a chemist. What would you do like on a day-to-day basis? Would you be working at GSK, for instance?
What would you be doing if you were working at GSK?
Yeah, on a day-to-day basis, what would your day kind of look like?
As a graduate chemist working in the lab, you would be responsible for making molecules. You would be doing this synthetic chemistry. You'd be hopefully contributing to the design of synthetic routes to those compounds. You'd be looking at the biological data and thinking of which molecules you needed to make next in the process. So you would be in the lab doing synthetic chemistry. The one thing I meant to mention and I forgot to mention, and I think it comes as an eye-opener to chemists. I'm a chemist by training. I've spent most of my career thinking much more about biology than I do about chemistry. And when I was at Belfast, I probably spent 95% of my time thinking about biology and biological aspects of drug discovery, and only 5% of my time thinking about chemistry. And I think that's an important message. If you want to be able to direct the strategy of drug discovery, you need to be able to talk to your colleagues in other scientific disciplines. So you need to be able to have at least a basic understanding of those other scientific disciplines so that you can interact with your colleagues in biology, in pharmacology, in drug metabolism, and so that you can then influence the direction in which science goes. If all you think about is chemistry, then you may find that your career doesn't really allow you to move much beyond working in the chemistry laboratory.
But is everything synthesized by this automated approach, or would you be like kind of doing like a little column yourself?
Well, I think automation was the big rage in the late 1990s, and SmithKline Beecham and then GlaxoSmithKline invested huge amounts of money in automation technology at that time. I think automation's still important, but I think that sort of initial flurry of high throughput combinatorial chemistry has gone. It's incredibly expensive to make and test all of these compounds, and particularly if the type of combinatorial chemistry that you're doing is not really enhancing your compound collection. If you're making thousands of molecules by combining carboxylic acids and amides and amines to make amides, are you really advancing your knowledge of the target? So I think I'm being disrespectful to some very, very good combinatorial chemistry there. Please don't misunderstand what I'm saying. But I think the sort of idea that everything has to be done combinatorially, that was all the rage in the 1990s. I think a much more sensible approach is being taken these days, and it's not about making numbers of compounds, it's about making the right compound. It's about understanding the principles and applying those principles to think carefully about the molecules that you make. It'd be great if you could do a drug discovery program and make the smallest possible number of compounds to get from A to Z. Wouldn't have to screen tens of thousands then.
Well, thank you very much. I'm sure there'll be lots of discussion afterwards with the refreshments, but if you just join me in thanking you very much.